Hello, I would like to tell you about what I did during my PhD. I work on multilayer networks, and I use them to identify clinically meaningful subsets of patients in diseases, COVID and sepsis. So we are living in an age of explosion of biological data. We have more than just transcriptomics. We have single-cell transcriptomics. We have proteomics, metabolomics, and detailed clinical information on patients. And to deliver the promises of precision medicine, we need to understand how to represent these complex biological data in a most efficient way. So the first question that I try to answer in my PhD is, how can we integrate multiple molecular modalities and data sets in a statistically rigorous way and with confidence? Second question is motivated more by the biology, and that's driven by the observation that a lot of diseases share a molecular underpinning. And even though they are classified as separate diseases by a lot of doctors, we now, with increasing understanding of molecular biology, we understand that they probably should be classified together. So I look at whether we can find subphenotypes of COVID-19 and whether there are similarities to sepsis. I work on a combat data set. We have over 100 COVID patients. We have convalescent patients. We have healthy individuals and sepsis individuals, from whom we took blood samples and then ran a number of modalities on these blood samples. We have cell composition data, site of 10x. We have proteomics. We have transcriptomics, also single-cell transcriptomics, and importantly, a lot of clinical information on these individuals. And the approach that I employ is I take all this data that we have, I create patient similarity networks, I then connect them into a multi-layer network. I identify clusters in that multi-layer network and explore what we see biologically there. The first question when you endeavor to do that is, how do I pick a network out of all the possible networks? So if you constructed a network, you know that there's a lot of parameters to choose, and it's hard to decide what is a good network. People often choose heuristics or approaches that basically, well, a correlation of 0.8 sounds like a good correlation. But a second question related to that is, how do we quantify information in modalities, and how do we pick which modalities are most useful in a given disease? And I explore that using Pareto cogent, and the intuition behind this method is that if we introduce small perturbation to the data, we should observe similar networks produced from this perturbed data if our method of recovering networks is good. So I start with a single modality, for example, proteomics. Patients are rows and columns are the proteins. And then we create feature resampled sub-data sets. So I say that some features are gray features, which will be shared. Some features are orange and some are blue. And so I create iteratively a number of networks. And when I create orange networks, I use the shared gray features and orange features to calculate correlation networks or similarity networks. And when I create the blue networks, I use gray and blue features. I then also create an expected graph, so something that we would expect under random permutation of the nodes. This preserves the various features of the graph, such as triangle inequality, but disrupts clusters. I do it a number of times. It's an iterative method. And at every step, I quantify the consistency and complexity of the pairs of networks. So I compare the observed and random network. I calculate the intersection and union graph. And what you can see here is the edges that are dotted and colored with purple or yellow. They are the ones that only occur in one of the networks at a certain threshold. The ones that are green and solid are present in both networks from the perturbed datasets. So we can do that for the random networks and or comparison of random and observed network. And then we see that there's not that much information shared between these two networks. But when we look at two observed networks, we see that a lot of edges are green and solid, so they are present in both subdata set generated networks. I can then explore how this varies when I change parameters of how I construct this network. So this is a problem that we are solving. How do I pick a good method? How do I pick the good parameters of a method that guarantee this intuitive property that I described earlier? So when we take very sparse networks, when we set this density to a very low value, we see that the edges that we select are on the left when compared to observed networks. They are actually quite consistent because out of the 10 edges, 4 of them are shared between the two sub-data sets. And in the random data set, when we are comparing with a random network, none of them are shared. But this consistency, while good, we don't have a structural complexity in the observed network. So we need to explore other parameters. And we can quantify these 2 parameters, the complexity and consistency, and then calculate the randomness adjusted complexity and consistency. When we go to the other end of the scale and we explore very dense networks, we see that, yeah, there is a lot of consistency and there's a lot of complexity in the observed networks. But there's also a lot of complexity and consistency in the comparison with the random networks. So this is something that, well, this comparison with random networks allowed us to discover that the very dense networks are fulfilling these properties just by the virtue of being very dense. So when we scan across a number of such parameters and when we go through the whole spectrum of densities that we can explore, we can identify this parameter, this threshold of density that leads to the largest difference between observed complexity and consistency and the value when it's adjusted for the random network. So on the left, we can see that at this threshold, the observed networks are both very consistent and structurally complex. They reveal these 2 clusters. And the network on the right, the comparison with the random network, leads to a very inconsistent network that doesn't really have much structure in the green subnetwork. So we can scan through parameters in this way. We can explore not just the density, but we can also explore different similarity measures. We can explore different ways of pre-processing data. And what our method offers you is a understanding of which parameters are leading to these complex and consistent networks, and also helps you quantify these properties. We can then pick the best networks. And when we apply this method on our actual data, for classical monocytes, we identify the network that is that the edges that we select are highly significant. So what we observe is that on the dotted line at the bottom, that's the random expectation across different thresholds of density. So the line gets more yellow as you get towards the top right, and that's a network with more edges. And the top line represents the observed data. So we see that at 350 edges in both, in the random, we have a very low consistency and complexity. In the observed data, we have a very high complexity and consistency. We can identify this optimal point by finding a point that the difference of these 2 curves is closest to the 1 point, which represents the optimal consistency and complexity. When we applied this to the whole data set, so over 30 modalities, we identified a number of networks, and they fulfill this property of being complex and consistent in this optimal way. And we also understand how they compare against each other. So when we look at these both parameters, we can plot them in the same space, and what this plot helps you understand is which modalities to pick when analyzing a specific data set. So C-monocytes and bulk RNA-seq and non-classical monocytes are the modalities that in the COVID data set, in an unsupervised way, provide the largest evidence for this clustered, so patient stratification nature of the data set, and they are also very consistent. And when we plot these two networks, so the bulk RNA-seq network and classical monocyte network, we can see that when we overlay the diagnosis information on top of these networks, we see that patients indeed cluster into this green healthy cluster, then expand into the red, more severe COVID, and then yellow, which is the sepsis group. And different modalities obviously reveal different types of information. The next step of this approach is to take these optimal networks and build a multi-layer network out of them. So we connect nodes by their identity across modalities. We weigh the modalities by this information, Pareto-Cogent-derived information score. And then we cluster this multi-layer network using Leiden algorithm adapted to that to multi-layer networks. And now I'll talk about the results. We identify in most of the analysis, we identify 5 clusters. This is using only the molecular data, but we are able to recover the diagnosis that these patients had assigned by a clinician. This is an enrichment plot of the different diagnoses in the different clusters. We also identify when comparing with regular diagnostic tests that are run in the clinic, we identify differential values of platelet counts, oxygen levels, and oxygen support needs, D-dimer, monocyte levels, and other features that are clinically important using only this unsupervised molecular information. And when we then explore what drives this clustering, we can identify features that are most predictive of our clusters and compare that to the features that are most predictive of the WHO, so clinical standard-based clusters. And we see that we focus on classical monocytes, cycling monocytes, and particularly on the interferon response B-naive cells. Our clusters also have significantly differential ICU-free survival. So within this hospitalized COVID group, we identify classes of patients that are more likely to be intubated. And then we narrow down on what is different about WHO and our classification. And we see that our approach prioritizes IL-33 inflammation, interferon response activation, and monocyte activity. And we do that by training 2 logistic regression models and comparing feature coefficients when features are clustered in correlated blocks. And in this way, we can identify the most significant differences between the 2 classifications. We also validate our approach on external data set. Here I just show a data set, one of these data sets. We validated it in a couple of data sets. And we see that when we create a score of severity based on our clusters, it separates patients that when you calculate this score on the first day, it predicts whether they are likely to die or to be discharged. Our scores are also predictive of a number of other clinical features such as D-dimers, oxygen levels, and so on. And the enrichments and associations that we found in our data set are also recovered in these external data sets. So in summary, I've showed you a method that takes you from molecular data from multiple modalities and identifies informative and reproducible networks, ranks them in terms of information that they inherently contain within your disease setting, integrates them into a multi-layer network, and identifies in that multi-layer network clusters of patients, so disease subtypes. These subtypes have distinct biomarkers that are different from WHO and are more predictive of clinical features of interest, and they are reproducible in external data sets. Thank you very much.
Thank you. We have time for a few questions.
Hi. I'm wondering if you compare your approach with building embeddings on your multi-layer network and then clustering the embeddings within class instead of Leiden. That's question number 1. And question number 2, would you be able to add another 4 category of COVID patients, long COVID maybe, to your data set and see if you can find, because I think long COVID is something that would be very interesting. network as well?
Yeah, so on the first question, we don't look at the multi-layer based embeddings. So this is something that definitely would be useful to explore, whether using the information, not just the graph information, but also the underlying molecular features information enriches this representation. And on the second question on long COVID, here I'm using a cohort that was that basically has been deeply phenotyped, and this recruitment has finished. But we can use external data sets, and there are long COVID data sets that could be included in this pipeline.
Maybe using embeddings would help with projecting the external COVID data set.
Yes, we see that in our convalescent patients. We do see a split of convalescent patients. So some of them, well, there is this difference between patients who seemingly recovered.
Rebecca, go ahead.
Thank you for the great talk. I find it very interesting how you identify the common subnetworks. So I have a question about that. What is the random graph model that you use as control, and how much would the networks that you identify depend on the random graph model?
Yeah, so this is something that, well, you can use models. What we used was a quadratic assignment procedure. So we basically permuted the node assignments. So it keeps the correlations intact. It just changes where they wire. Yeah.
Time for another question or two. So I had a question about the impact of, so you have this approach where you dial the threshold, I think,
Yeah.
to optimize the difference in complexity. Did you evaluate the impact of that step? So if I don't have that step, the impact on the downstream clusters and the feature derived from those clusters, does that have a major impact? Or can you talk about that a bit?
Yes. So basically, by varying this threshold, we choose the optimal networks in the sort of enrichment of a random networks, right? So if we chose another threshold, because we have to choose some threshold when we are creating this correlation or distance matrices, we can see basically where it would lie on that line and how much was over the random comparator it would be. Further down the line, well, the networks that you input into the multi-layer network algorithm, they influence how many clusters you get. So yes, we did a lot of ablation studies on how various parameters influence that, and yeah, it's a complex story there.
Thank you. Maybe one last question. Okay, if not, let's thank the speaker. Thank you.
Thank you.